Today we will be discussing antifreeze protein properties in Antarctic notothenoid fishes. Notothenoidae is a perch-like suborder of fish that primarily includes Antarctic fish. These fish are able to survive in sub-zero temperatures while avoiding the cell damage caused by freezing. This is speculated to be caused by the production of antifreeze proteins, or AFPs. AFPs are a class of polypeptides that function by binding to ice crystals and inhibiting their growth. AFPs are likely essential for the survival of fish living in Antarctic waters. Cell freezing and eventual thawing causes the formation of crystals intracellularly and/or extracellularly. The crystals can destroy the cell membrane, compromising the cell's integrity, and can be fatal to the organism. This is shown in our figure 1. AFPs work by creating a non-equilibrium freezing point that widens the temperature gap between the melting and freezing points, decreasing the temperature at which ice forms. This phenomenon is called thermal hysteresis. The presence of AFPs are responsible for the variation in ice morphology. Ice normally forms in a bulky three-dimensional crystal shape, but AFPs cause the formation of two-dimensional sheets. This is shown in our figure 2. This study examines the specific functions of AFPs within notothenoid fish, such as how they are able to prevent cell death while submerged in freezing waters. Figure 1 shows the impact of different freezing processes on cells. Cell freezing causes the formation of ice crystals, which can form outside or inside of the cell in response to thawing. The location of ice crystals is determined by cellular thermal history. The crystals can destroy the cell membrane, compromising the cell's integrity, and can be fatal to the organism. This is important to note because the presence of AFPs prevent ice crystals from forming and does not compromise the cell's integrity. Figure 2 illustrates the general process of how AFPs work. Ice normally has an EQ-FMP that forms ice in bulky 3D shapes. AFPs create a non-equilibrium freezing point by widening the temperature gap between the melting and freezing point and forms ice in a nearly 2D sheet. The ice morphology variations shown in Figure 2 are due to the presence of AFPs. Figure 3 shows the location of the notothenoid fish that were used in the following research. The map shows where nets were cast, field expositions, and temperature logging for the study. Figure 4 shows the recrystallization properties of solutions with and without AFPs. The method used to collect this data was an ice recrystallization inhibition assay of 30% sucrose in a 1:1 ratio via warming solution. The presence of AFPs in this solution shows the inhibition of ice growth. The solution without AFPs shows that after 60 minutes in a -6°C stage, large ice crystals were formed. After 60 minutes in the same conditions, ice crystals were not observed in the solution in which AFPs were present. This demonstrates how thermal hysteresis is able to inhibit the growth of ice crystals. More specifically, it lowers the freezing point, allowing water to remain in its liquid state in colder environments. Figure 5 shows the effect of water temperature on the fraction of fish with internal ice. This data was collected by recording temperature profiles at Cape Evans on January 3rd and January 10th. Fish were then collected on the same days from 17 to 30 meters below the surface. Graph A shows the temperature of water compared to the depth for the two different dates. Graph B shows the ratio of fish that exhibited internal ice. It is important to note that despite the water temperature increasing between January 3rd and January 10th, the fraction of fish with internal ice remained generally constant. This shows how irreversible the reaction between ice and AFPs is, and how it persists even when the water temperature approaches non-freezing levels. Figure 6 shows the difference between actual and predicted freezing and melting points within notothenoid fish. Light microscopy and a freezing point osmometer were used to collect this data. For this figure, it is important to keep in mind that EQ-FMP stands for Equilibrium Freezing Melting Point. The upper portion of panel A shows the difference from EQ-FMP in the blood of 5 different species of notothenoid fish. The lower portion of panel A shows the difference from EQ-FMP in the bile, extradural fluid, intestinal fluid, urine, and vitreous humor of the T-pen fish species. With this information, we can conclude that most notothenoid fish follow a narrow range of EQ-FMPs at around -1°C. In panel B, fish in purified AFP solutions were put into ice at temperatures above and below their EQ-FMPs. The upper portion of panel B shows the difference between the actual freezing point and the EQ-FMP, and the difference between the actual melting point, in the EQFMP in the blood of 5 different fish species that contain AFPs. The lower portion shows the difference from EQFMP of purified AFP solutions. From this data, we can conclude that fish that exhibit AFPs are able to withstand lower temperatures because the freezing point has decreased. In conclusion, the presence of AFPs in bodily fluids of Notothionoidae fish prevents harmful ice crystals from forming. Thermal hysteresis is able to inhibit the growth of ice crystals, and more specifically, it lowers the freezing point, allowing water to remain in the liquid state in colder environments. The reaction between ice and AFPs within the fish is irreversible, and it persists even when the water temperature approaches non-freezing levels. Future directions of AFP research are very broad. They are already being used in commercial applications of fruit and agriculture preservation. Some ice cream companies even use antifreeze proteins in their products to help maintain its consistency. AFPs could potentially be used for tissue and organ preservation for transplant operations. This would allow surgeons to have more time between transporting and operating without the tissues becoming unviable. However, one main question we have is if climate change will affect these fish. Will AFP levels in Antarctic fish decrease due to global sea warming? I guess we'll find out in a few years. Thank you.